determined. So we'll talk now about the graded elements. I'll explain what's going on with your Canvas site. And, this being a lab class, the first day we usually would go over lab safety. Obviously, there's no point in doing that when you're not in the lab. So we'll postpone that till when we are in person. Hopefully that's, by the fourth week we'll be back in business. But stay tuned. As I keep saying in these things, the hospitalization rates are going up and the case counts are in the 3,000 and 4,000 range on a daily basis. So I'm optimistic that we'll be back, in the first week of February, but let's not worry about that. Let's make the best of what we've got. And so, simple thing we can get rid of, we don't have to talk about lab safety. All right? And so what we will talk about here is just getting you started. So we're going to introduce you to the lab sequence, and I'm going to give you a quick rundown of the things I've got planned for us. And then we'll conclude with what I want you to do this week, and we'll move on with our day. So the Canvas site has completed syllabus, so go ahead and take a look at that. Usually the highlights that you're taking a look at are how's my grade based, right? And so the elements that we're going to be getting a grade from are, first of all, you're going to have a notebook, right? This is a lab class, so of course you have a notebook. I have probably about 3 years ago, we started using Microsoft OneNote as a digital lab notebook. I have lots of resources on the Canvas site about notebooks in general, and so I'm gradually updating them to the digital platform. But the reason I like OneNote, first of all, it's free. Okay, so you can, if you're a Microsoft person, you already have access to that on your PC. But if you're a Mac person, you can get OneNote through the Apple Store, and off you go. You can access it in the cloud, or you can access it off of your computer. And it also comes with Microsoft provides an app. I think it's called Office Lens. I'll have to double check. But that really works well for us. So I don't know how well you're going to see this, but the way I view, lab notebooks, I'm pretty old school, right? So, my lab notebook is usually one of these composition books. I write everything in here. And you can continue to do that, and I actually encourage you to do that. I also sometimes just write my notes on pieces of paper, right? And that's okay. What I would point out then is that how do you get it into your digital notebook? Old school, well, I would just shove it in there and off I would go. And obviously that's not something I could turn in a lab class, right? But nowadays, all I need to do is take a picture of it. And so the app I was talking about, Office Lens, allows you to scan your documents directly into your OneNote digital lab notebook. And then you're good to go. So my workflow is that when I'm in the lab, I don't drag around my computer on wet lab days. I just bring in my composition book and I write my notes. And then later, I'll scan my document and put it into the digital notebook and add some text and maybe some pictures and whatnot. So one of the things I'm going to do this semester that's new is I'm going to provide y'all with a template, because one of the things that happens is that they're sort of always like, well, what do I put in here? And so, well, you put in your work, right? And it's the purpose of a lab notebook is that it's the evidence that the work was done. And so I treat notebooks a little bit like a journal. Next week, hint, hint, we're not going to meet for lab. Martin Luther King holiday is on Monday, and so several of the lab classes are not going to meet. And so I'm going to set you up with a bunch of things to do for the following week, right? And so I want to spend some time working on the Canvas site. And so I thought, you know what? We're in the middle of a pandemic. We deserve to sort of figure a way out of this as easy as possible. So one of the things I'll do is I'll work to provide templates, because what goes in the notebook, it's a very standard kind of expectations, but sometimes it's hard to figure out when you're doing some digital work. So some of our bioinformatics work using BLAST and other bioinformatics tools, I tend to find students don't put much of anything in the notebook, because it's like, well, I'm doing it on my computer, right? And so maybe I'll get a screenshot or something. So anyway, so we'll help you with that. So the notebook is an important part of your graded element for the lab class, of course. And so the usual way this works is that your first part of the notebook is to create it, and then share the URL address with me. And then the additional thing is, I really don't want you to send me the hundreds and hundreds of character long URL for your OneNote. I want you to take the next step and create what's called a tiny URL, a shortened URL. And there's a variety of ways to do this. A long-standing approach is there's a guy that's run this site called tinyurl. It's easy to do. And so when you post your URL in Canvas for me, I'm going to ask you just share the tiny URL, right? And then what I will do is I will double check that I have access to your notebook. So when you share it, make sure that you allow me to edit it, okay? And this first step is due by next week, and it's worth 5 points, right? So your total notebook is worth 50 points. Right off the bat, 5 points just for sharing this thing. And obviously I will check, but let's get that taken care of, okay? The next thing that'll happen is by sort of mid-semester, I will be checking your notebook for completeness, and I'll give you a grade based on up to just a maximum of 5 points about how well you're doing based on the rubric that's present in the Canvas shell. And then at the end of the semester, I'll check it again. So one of the interesting things that happened is that I was getting emails periodically going, oh, can you delay when we're going to? No, I'm not delaying it. I already have access to it. So part of the job of doing this notebook is just to keep it up to date. Okay, so we'll help you figure out when to do it, but get it started, and we have a couple of weeks to sort of get up to speed on that. Okay, so the other thing, I'm not going to do a lab exam, but I am going to have a series of small quizzes based on the various things that we're doing. The quizzes are supposed to help you with the concepts. Some of it will be very specific, some of it's general, and I think there's about 10 of them. I forget off the top of my head. It says 10 right there, so I guess there's 10. Okay, the other thing, and so that'll be administered via Canvas. What about your notebook for that? Well, I would expect you to write in your notebook, when did you take the quiz? Right? It's part of the lab. Everything that you do for the lab shows up in your notebook. All right. The other thing, worksheets. Now, these aren't going to be things we hand out, right? They'll be administered via Canvas, but the various exercises that we do come with questions or things that you need to do. And so, yeah, I want that all put into your digital notebook, but to help you keep on track for some of these projects, which can take several weeks to complete, I've broken them down into, mini projects, right? So you do this, submit that. Do the next thing, submit that. And so a cumulative effort, and it allows me to see where things are sort of slowing down for you. It allows me to then help you troubleshoot the exercises that you're working on, okay? So some of them require directly submitting to Canvas. It says all. I forgot to write. All of it goes into your notebook, of course. It's evidence that you did the work. Okay, then finally, in the past, I've had 2 reports, but this is pandemic, so the heck with it. We're going to do just 1 report, and I'll talk a little bit about that. I'm working on figuring out how to sequence the lab. So I thought rather than giving you this timeline, since again we're not going to meet next week, what I'm going to do is kind of give you the general sense of the things that I'm planning for you to do. So basically wet lab versus dry lab. You know, wet lab you can expect to be running centrifuges and doing hot plate work and chopping stuff up, and certainly get those pipetters going, right? And there'll be protocols to do the various things. And so I thought, given that this is genomics, a really simple thing, but an underappreciated task is to get karyotypes. And so what we're going to do is I'll develop this or publish this protocol for basically cell smashing and getting chromosomes so that we can see them under the fluorescent microscope. So that's the DAPI imaging. And this may take us a while to do. It's simple, you know, in concept, but the actual, you know, getting it all to work and whatnot can be a little bit tricky, and that's okay. But the idea is that we can get this to work. You'll end up getting some brilliant photographs of your work. And so, you know, thinking about going forward, you're thinking about maybe a digital, folio or something, right? And so this would be good stuff to have in there. The other thing we're going to do is we didn't get done in the fall, but we're going to try again. So we're going to get our expression profiling mechanisms up. We have our primers for the tomatoes. We're going to add in some yeast. So the idea is we'll do, well, with the tomatoes, we've got 3 different strains that we can work with. So we can simply, you know, smash them up, extract the RNA, and then all things considered, we'll be able to get an expression profile to see what messenger RNAs are being present or being expressed in the 3 different color morphs. We'll also finish up our growth analysis. So the idea is to grow and plate yeast in different carbohydrate sources and potentially different stressors. So for example, exposing them to copper sulfate, which is used as a it's really funny, but this is called an organic approach to gardening. So the idea is that you mist your plants with copper sulfate, and that inhibits mold, right? Okay, so that's great, but you do realize that, like, dumping copper into the environment is not exactly an ecologically cool idea. And so at any rate, though, the other thing that happens is that the more that you administer herbicides and pesticides towards your pests, what do you think happens over time? The target evolves. And so yeast is a really good model system for these kinds of studies because, of course, yeast doubles about every 48 hours. That's a generation time. So we can witness response to selection, say copper sulfate, in the course of the semester if we get our stuff going. So that means there'll be weekly things to do to manage the yeast cultures. I choose yeast rather than bacteria because yeast is pretty forgiving. As long as we can avoid contamination, we can grow things at room temperature. We don't have to provide them with carbon dioxide, 5% carbon dioxide, like what you have to do with mammal cells. And since we can do them at room temperature, we don't have to worry about maintaining incubators, which you do with your bacteria often, okay? But the same kinds of attention to detail about how to manage cultures is very much applicable to yeast. So the goal is to teach you how to do, how to run a culture lab, right? So we're going to break that down. We're going to teach you about the autoclave and all of the other things, how to make media, how to do tests for contamination, and how to grow things in lab environment. And the payoff of this is that we'll be able to learn some simple little techniques about lab culture work, but also the meantime, you'll have yeast that are being exposed to different things, and so they will be evolving. And at the end of that, we'll have the start cultures, and then we'll have the end cultures. And so we have a perfect scenario for doing expression profiling and potentially sequencing to see if evolutionary changes occurred. So the expression profiling is done in a couple of ways, but we will focus on doing what's called quantitative PCR or qPCR, because we can do that in-house. We've got all of the instrumentation that we need. If things work out, we will also be identifying colonies that have evolved, and so that's potential for doing some sequencing work. And so I'll introduce a quick and dirty way to compare things by doing what's called high-resolution melting analysis. Now this last one here, population genetics of cat colony, you are aware that there's now a substantial effort by Dr. Grabowski and the environmental science and studies programs to sort of take care of the cats. And so we've talked in the background about, gee, it would be nice to see if we can do a pedigree, basically, to see who's related to whom. If you look at the faces of the cats, you can see some individuals that are definitely related to each other. There's a few cats that have sort of a very narrow pointed faces, and certainly coat colors. You tend to see, yeah, I'll bet you they had kids together, right? But this is something that we've been talking about, and so while it's not specifically a genomics project, it does work into the idea of we'll be selecting different markers that we wanted to use, and so that gets us into thinking about what's in the genome that can be used to discriminate among individual cats. Obviously, we have to figure out how to get samples, right? And so there's a variety of things that you can do, ranging from the you don't bother the cats at all, but you pick up their poop, and it's like, yeah, I'm not going to do that. And so the other thing is to do it from hair samples while they're trapping the cats. And so we just need to think about it a little bit. And so, yeah, that's part of it. So those are the wet lab components. Well, there's only 3 things listed here, right? But each one of these can take several weeks to complete. And so that's why I don't want to push us into these things until we sort of know where we're going to be. If we're going to continue to be online, that's fine. We will give you a good experience, but clearly we'll have to treat the wet lab component a little bit differently. Just to give you an idea how we would manage that is that we would bring you in small groups at a time to run some of the labs. So, we've got your back. We can figure this out. But right now, the instructions are to stay home, stay away. So we're going to honor that, okay? Okay, so then up at the top, look, genomics is really about sequence. And sequence you don't do in the lab the size we have. What you do is you extract DNA or RNA, and then you send it off to a core facility somewhere, usually on the mainland, and then they send you back sequence information, right? And so then what you do is you spend your time analyzing the sequence according to your experimental design and other kinds of questions. And so I've done this now for several years. I've developed what I think is a pretty interesting series of exercises that address the question. So look, we have information about single nucleotide polymorphisms in genes. These are available in databases, and so we query a database that's all about humans, right? And so we can look in different conditions in humans, ranging from various kinds of heart problems to schizophrenia to alcoholism, you name it. And so you pick a phenotype, a complex phenotype, and then we use databases to pick through and find the information in which they've compared 2 or more groups, those who have type 2 diabetes and those that don't. And so they did expression profiling. That was one thing, but then they also grabbed sequence information. And so what you're looking for are single nucleotide differences that are found only in your type 2 diabetes group, right? And not in your control group. And that's what the database is. Now you should appreciate, just because you find a difference doesn't mean that that's why somebody has type 2 diabetes. So I've developed, based on my background in evolutionary biology and whatnot, is this pretty interesting approach to using evolution as our variation generator and to test hypotheses about whether or not the gene that's been identified through this S&P analysis is something that you maybe want to consider. Yeah, that's evidence that there's something really going on. So it doesn't make a lot of sense, I know, but bear with me. I have a workflow that explains the steps that we're doing, and I also, of course, have a whole bunch of background in the course website to explain why this is. And we've got some time, right? So not next week, but beginning in the third week, we will address the science part of questions here, as well as the how to do things. Okay? And then the other dry lab thing is, of course, we will continue to teach you how to do primer design. Since you're going to be doing PCR, you have to make your own primers, and so we'll show you how to do that. That too is a dry lab. It's all done with software. Okay, so I just sort of stepped through the bioinformatics project, and so this is kind of the outline. Some of the things that you're going to be doing are you're going to ask questions about that database. It's up, and you're going to take. All right. So the project at the end product is you're going to put together a PowerPoint and then produce a video. I've been doing this now certainly through the pandemic, and I just find that the easiest thing to do is to have you sit at home and do a presentation and record it, and then we publish the video, and then as class you can independently watch the video, and I do have you do a peer evaluation of the product. Okay? So what do you do this week? Go to Canvas, if you haven't already done so, and find the instructions about setting up the digital notebook. Set up your digital notebook. Share it with me by creating sharing the URL. Go to the tiny URL, make it short, and then posting that to the Canvas site. I'm also asking you to take a short survey. Really, all I want to know is what kind of computer you're using. And if you don't have a computer, well, I need to know that too. So I have you download and install some software to make your life much, much easier doing the bioinformatics. But it is possible to do things without having a laptop or a desktop. But I need to know that so that I can help you. But so then the other part is that Macs and PCs are different enough that I need to know who's out there with what kind of computer. That's part of the troubleshooting thing that we need to do. Is you always got to tell somebody what kind of computer you're working with, and you say, oh, well, it's a Mac, and it's like, okay, well, I have a MacBook too, but it is now no longer, I can't upgrade it past their Catalina operating system. So that's now been over a year and a half since I've been able to upgrade, because the computer hard drive is too small, so it can't run the newest version of operating software. Computer's perfectly fine, but it's not the latest and greatest, right? And so it's different from a MacBook that you bought 2 weeks ago. And then the other thing Apple did is that they are using their own CPU, their own processor. And that's cool because it's really, really fast, and it's really good on your battery life, and that's excellent. But most of the software expects you to be using Intel chips, And so there's a big shift between that. And so manufacturers, software vendors out there are scrambling to sort of produce things for the new version. So anyway, I need to know that, sort of what operating system you have and what your CPU is. And it's just a survey, but get it done as soon as you can, and they'll give you 5 points bonus for lab, and why not? Let's give you 5 points bonus for the lecture as well, because of course we'll do some of this stuff in lecture too. So let's try to incentivize that so that I can get that information, and that will help me as I'm preparing videos and fact sheets for the work to come. Okay, so again, Note Lab next week. On the one hand, I apologize for that, but on the other, it's like, look, we're all swamped right now trying to get everything going. I've had to relearn how to do this lecture by streaming things, and we've just got a lot on our plates, and so rather than spend hours and hours and hours to build things that in the end we may not need, let's go ahead and take advantage of the holiday, and so we'll get together again in week 3. Okay, so I'll be busy behind the scenes, but you don't have to be. Just take care of those 2 things, the set up the notebook, take the survey, and I will see you regularly in class for lecture, right? Regular lecture schedule. Okay, that's all I got. So I will sign off now, and thanks for listening.